sample of Sue's lymph fluid was sent off to the lab in order to identify the pathogen that's making her sick. You'll need the sequence of the pathogen's DNA to determine its identity. You will help complete this analysis, but first, let's investigate how DNA sequencing works. DNA sequencing is how scientists determine the precise order of nucleotides within a DNA molecule. Many different methods are used to perform this process, and even now, next-generation sequencing methods can sequence an entire human genome within a single day. For this presentation, we'll focus on a common method used to sequence DNA for clinical purposes, called cycle sequencing. Similar to PCR, cycle sequencing uses a thermal cycler, but there's a twist. Instead of producing multiple identical copies of the template DNA strand, the goal of a sequencing reaction is to produce a set of DNA fragments that differ in length from each other by a single base. The fragments are then separated by size, and the bases at the end are identified, recreating the original sequence of the DNA. Let's look at this process in more depth. First, let's zoom into a segment of DNA from the pathogen that we just copied using PCR. To begin this sequencing process, this double-stranded DNA segment is denatured, meaning it's separated into two strands. We call these the template and complementary strands. The template strand is then mixed with primers just like the ones used in PCR. DNA polymerase, the enzyme that assembles nucleotides to create DNA. Free nucleotides, which are the bases adenine, thymine, cytosine, and guanine, and fluorescently tagged free nucleotides. These are A's, T's, C's, and G's that glow when run through a laser. Each one is labeled with a specific color fluorescent tag. For our reaction, the A's will be fluorescent green, the C's blue, the T's red, and the G's yellow. As the reaction runs, the creation of each DNA fragment starts with the primer. After the primer attaches to the DNA template strand, the DNA polymerase goes to work and begins to add the complementary nucleotides to build the DNA. For example, whenever there's an A in the template strand, DNA polymerase adds a T. Whenever there's a C in the template strand, it adds a G. The DNA polymerase continues to follow the base pairing rules to add nucleotides to the chain. Whenever a fluorescently tagged nucleotide gets added to a chain, the DNA polymerase stops, and that chain is complete. This happens randomly, creating fragments of varying lengths. Because there are billions of free nucleotides available in the mixture, fragments of every length will be created. The reaction stops when there are no longer nucleotides available. The result is that each DNA fragment created is a specific length and always ends with a fluorescently tagged nucleotide. Now that you have all of the DNA fragments, you need to separate these fragments by size. Imagine how time-consuming it would be to separate these sequences by a traditional gel. Luckily, that's where our automatic sequencer comes in. The automatic sequencer performs gel electrophoresis to separate the DNA by size and then analyzes the results. Notice the thin capillary tubes inside the sequencer. The tubes are filled with a buffer solution, and the fluorescently tagged DNA pieces are inserted into these tubes. Let's look at what happens inside one of these tubes. An electric current is applied to the tube so that the negatively charged DNA will move down the tube to the positive end. The shortest segments of the DNA move through the tube the fastest, so they reach the bottom of the tube first. Near the bottom, a laser beam passes through the tube, making the fluorescently tagged nucleotides glow as they pass through it. Optical detectors determine the color of the fluorescence and relay this information to the computer. The color of the fluorescent tag identifies the nucleotide at the end of each fragment. Remember, red is thymine, blue is cytosine, yellow is guanine, and green is adenine. Because yellow can be difficult to see, the computer converts the yellow to black. The fragment of DNA that reaches the bottom of the tube first is the smallest fragment. We can determine that this fragment represents the first nucleotide in the sequence. In this case, the first nucleotide is thymine. Because of its fluorescent tag, it will show up as a red peak. The next fragment to pass by the detector is the next smallest fragment, representing the second nucleotide in the sequence. In this case, our second nucleotide is guanine and will show up as a black peak. As the detector identifies each fragment, we're able to piece together the nucleotide sequence base by base. For our sample, the sequencer generated a sequence of T, G, G, C, A, T, C, A. This is the complementary sequence to our DNA template strand. This process has allowed us to identify the exact DNA sequence in the sample. Sometimes it's difficult to interpret a nucleotide peak, as there are places where the peaks may overlap. This is particularly common at the beginning and end of a sequence. These ambiguous places are often labeled with an N rather than one of the 4 nucleotides. Now that you've learned how DNA sequencing works, it's time to sequence the DNA of the pathogen making Sue sick. The sequencing preparation has been done for you, and the sample was run through the sequencer. Now it's your job to analyze the sequence data and determine the exact DNA sequence. Once you have the DNA sequence, you can use bioinformatics tools to identify the specific organism. Good luck!